It's not turned green yet. Okay.
It's him, so.
Okay. Hello. Welcome to today's live webcast on a good death. Are our euthanasia methods for lab animals actually humane? My name is Dr. Stephen Wickens, and I work for the UK charity, the University's Federation for Animal Welfare, and I'll be your moderator for today's event. The educational web seminar is presented by LabRoots, and for more information about this organisation, visit labroots.com. A few important announcements before we begin. Firstly, this webcast is designed to be interactive, and we encourage you to ask questions about the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window, and we'll try and answer as many of your questions as we can. And if you wish to enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. Now, if you're having any technical problems, either viewing or hearing this presentation, please click on the support button on the top right of your presentation window, or submit your problem through the green Q&A button lower left. Okay, I would now like to introduce you to today's speaker, my colleague, Dr. Huw Golledge. He is the Senior Scientific Programme Manager at the University's Federation for Animal Welfare and the Humane Slaughter Association. Until recently, Huw was a researcher at Newcastle University in the UK and is a current member of the UK government's advisory body, Animals in Science Committee. Huw.
Steve, thanks. And I'd like to say hello to everyone who's virtually listening, and thanks to Shapan and the rest of the organisers for inviting me to give this talk. As Steve said, I'm going to be talking about whether euthanasia methods for lab animals are actually humane. In other words, do we give these millions of animals that we use in science every year a good death? Because the word euthanasia actually means a good death. And that's typically taken to mean a death which occurs without pain, suffering or distress. So do we know if they actually have a good death? And if not, can we work out how to give them a better death? And at the start of these talks, I usually resort to a bit of semantics, because you'll notice through most of this talk that I'm going to use the word killing rather than euthanasia. And that is because both euthanasia means a good death, which means a humane death, and I'm not certain that the death of every lab animal is humane, and also because it's sometimes taken to mean a death which is brought about to end suffering when we think about companion animals being put to sleep or injured animals. And lots of lab animals are killed for reasons other than to end their suffering, because they're just control subjects at the end of a study, animals that have reached the end of a study, overstock at the breeders, and therefore I think it's more appropriate to use the term killing, but I do understand that lots of people prefer or are happier with the term euthanasia. So the broad aims of what I want to do today are to consider the welfare impact of the way lab animals are killed, taking rodents as my example, both because we know the most about them, I think, and because they're some of the most common animals in labs, and really just to question whether we understand the harms of killing them in terms of whether they suffer when they're killed, and whether those are adequately represented in harm-benefit analysis, and if not, suggest what we might do to improve things. So there are lots of ways to kill animals, and I'm sure you've heard of lots of these methods: cervical dislocation, decapitation, all sorts of physical ways of killing animals, and chemical ways, drug overdoses, poisons, and so on. How do we choose from this great plethora of methods which ones we should use for lab animals and lab rodents specifically? And I think, obviously, as an animal welfarist, that the primary concern should be the welfare constraints. In other words, choosing the most humane method, the method which causes minimal pain or distress. And if distress or pain is unavoidable, we may want to think about using the quickest method just to limit the duration of any of these welfare impacts. And as a secondary concern, perhaps we should also consider other animals that are in the presence of animals being killed, because although the data is not unequivocal, there are suggestions that animals shouldn't witness other animals being killed because it may distress them. we shouldn't forget, of course, that we are using these animals for scientific purposes, and so there are scientific constraints quite often on the way we kill them too. If you want samples of the animal's brain to do anatomical or histological examination, you probably don't want to kill the animal by a physical method that causes trauma to the brain. On the other hand, if you want to sample certain tissue or blood, you may not want it contaminated with drugs or other chemicals, and there are situations where you may need to arrest a physiological process at the time of death, which may necessitate a method that causes a very rapid death. And perhaps least palatably, there are practical constraints. In the real world, cost is an issue. Some of the equipment or drugs that are used are expensive. There are also constraints based around the number of animals. It's not uncommon to need to kill tens or hundreds of animals in a day. You can't really ask one person to do that one by one using a physical method, so methods where you can kill more animals or where there's some form of automation may be preferred. Availability of equipment and safety are also concerns. Some of the things that are used to kill these animals are not particularly safe, and if you don't have the appropriate precautions in your institution, you probably can't use them. And finally, something that's often overlooked is the effect of killing these animals on the people who do it, and people often find certain methods more unpleasant than others. We shouldn't be asking people to constantly carry out methods that they find unpleasant. That clearly shouldn't dictate which method we use if it happens that an unpleasant looking method is actually more humane, but it's definitely something we need to think about. So, having talked about all those constraints, really what I'm concerned with is welfare. So my question is really, how can we tell if the common methods we use for killing these animals are humane? And as someone who's tried to work on this, I'm going to claim, like all scientists do, that the thing they work on is uniquely difficult. But actually, I think this is quite a difficult question, animal welfare, because several of the routes you have to ask questions of animals in other welfare questions are not available to you. We can't ask an animal that's just been killed what happened to it, but you can indirectly infer what happened to an animal that had something painful occur. Similarly, we can't use anthropomorphism because very few human beings know what it's like to be killed by all of those gruesome methods that I told you about. But in other situations, we can assume that if a procedure is painful to humans, such as a laparotomy or breaking your leg, that it's probably going to hurt a rat as well. And finally, lots of the things that are used to kill these animals in terms of drugs are agents that interfere with their behavior in a way that may preclude them from expressing signs of poor welfare, sedative drugs and so on. So there are a number of problems, but despite that, I'm going to argue that there's been some really good progress made lately, which does tell us quite a lot about the welfare impacts of some of the common methods used to kill these animals. And what I'm going to do is go through some of the commonly used methods and just discuss some of the welfare issues concerning those methods. And I'm going to start with the physical techniques, which are really cervical dislocation, concussion, and decapitation, which I've put in brackets because, at least in Europe, there's a assumption that decapitation should really only be used when there's a very strong scientific justification, but nonetheless it is used, so it's worth considering. And in the case of cervical dislocation, breaking the neck, and decapitation, there are a couple of welfare concerns that really come up. The first is that although it's sometimes assumed that these methods effectively kill animals instantaneously, there's some evidence to the contrary, and there's a bit of data here in this graph on the left with the red trace, which is an electroencephalogram, the brain activity in a rat which was decapitated at time 0 on that graph, where there's a big vertical spike in the data. And what you'll see is that there's ongoing activity after that event. Now, this animal was anaesthetized, I should say, so it's not suffering. But what Kongara and colleagues infer from this is that there's a possibility that this animal would not have lost consciousness instantaneously had it been a typical animal undergoing this procedure. So there's a concern that these animals might have a few seconds where they have the potential to suffer, and it's even suggested that there are signatures in that brain activity that might correspond to nociception and therefore potentially to pain. Sam Kartner and colleagues have done similar studies looking at cervical dislocation, and similarly it doesn't appear to instantly abolish activity in these animals' brains. So that's something of a concern about these methods. Alongside that, there's a sort of procedural issue with cervical dislocation, which is that some studies find that there is a significant failure rate when people attempt cervical dislocation in small rodents. Again, this was done in anaesthetized animals, so no animals suffered when this data was obtained, but this data from Larry Carboni's lab looking at people attempting to do cervical dislocation in anaesthetized animals suggested a quite high failure rate, and by failure we mean that the animal kept breathing, probably either because there was no dislocation of vertebrae or vertebrae in the thoracic rather than cervical region were dislocated. So there's a sort of procedural problem there which may be surmountable by better training, but it's definitely a concern. And finally we have concussion, which has sort of dropped off the radar a bit. It's not talked about very much for lab rodents, but it might be the best physical option, because what this involves is hitting the skull of the animal, or more typically picking the animal up and hitting it on a hard object. And it should, in theory, render the animal instantly unconscious, not necessarily dead, but unconscious, effectively stunning it. And therefore, it could be one of the more humane approaches done right. And of course, this is a method that we're really quite comfortable with when we're talking about slaughtering livestock, where captive bolt stunning of large ruminants and so on is commonly accepted as a method to stun them before they're killed. So there's very little experimental data about rodents, but I think it's a method that people should bear in mind, especially when dealing with small numbers of animals. Oops. So we come on now to some of the chemical methods and to an overdose of anaesthetic, which is another commonly used way to kill lots of species. Pentobarbital is the most commonly used agent both in labs, and it's also what veterinarians would use to put to sleep your cat or dog, quite likely, if you take it to the vet at the end of its life. The difference with rodents is that typically this drug has to be given by the intraperitoneal route, whereas in larger animals it can be given intravenously. And that actually raises a few welfare issues of its own. First of all, the drug itself, or the solution, is quite alkaline. There is potential for pain on injection, which is manifested both as signs of abdominal writhes and as activation of nociceptive pathways. And IP injection also means there's a much longer transition into unconsciousness than by the IV route, and we're not quite sure how these animals feel during that long transition to unconsciousness. I should point out that this Svensson paper that I've referenced, and if you can't see the reference, please do email for these references at the end. This Svensson paper does also suggest a possible solution to ameliorate the pain on injection by adding lidocaine to the solution. So there is potential to ameliorate that. Okay, so I'm going to move on now for a large bulk of this talk to looking at exposure to inhaled substances, gases or volatile agents for killing these animals. And really the ones that I'm going to consider of a larger group are the most common ones, which are carbon dioxide, inert gases like argon and nitrogen, which simply kill these animals by displacing the air from the chamber that they're in and therefore allowing them to asphyxiate, and volatile anesthetics, drugs like isoflurane and sevoflurane, which are commonly used for surgery. And these drugs don't necessarily kill the animals, they're quite safe, that's why we like them as anesthetics. And certainly if you deliver them from a vaporizer, it's going to take you a long time to kill a box full of mice with isoflurane. But they can be used to render animals safely unconscious. and then allow you to use another technique, carbon dioxide or a physical method. And of course, when you're killing animals, you don't necessarily have to deliver these anesthetics by a vaporizer, and that may allow you to deliver much higher concentrations, which could be lethal. So going back to carbon dioxide, this is by far the most commonly used way to kill rodents in labs, as well as it's used commonly in a number of other species. It simply involves the animal breathing carbon dioxide in a chamber until it loses consciousness and eventually dies. And it's popular because it's simple and safe. It does what it's supposed to do. It's cheap. It doesn't contaminate tissues, which means you can either do sampling of those tissues, or the carcasses of animals can be used for food for other animals, which is a significant consideration in some cases. And some people find this much less distressing than some of those other methods we've talked about. But concerns about the welfare of this agent have dogged it for years, decades. This paper here is 20 years old now, raising concerns about whether it causes pain and distress. So I'll try and tackle those two questions, whether or not carbon dioxide causes pain, whether or not it causes animals distress by some mechanism other than pain, and throw in a third question, which is simply, is there something else that we could use now in terms of an inhaled agent which would allow us to comfortably replace carbon dioxide if we have these concerns? So the pain issue is a pretty easy one to deal with. Carbon dioxide reacts with water to form carbonic acid. That will happen anywhere where there's water present, including on moist tissues in the bodies of animals which are exposed to CO2, so the nasal passages, the corneal epithelium, the respiratory tract, and so on. And that liberates protons, which can activate nociceptive neurons. Acids cause a stinging sensation, we know that. And circumstantially, there's evidence to make us think that it's pretty likely that exposing rodents to a high concentration of CO2 would cause pain. First of all, it hurts humans. If you breathe 50% CO2, it will start to hurt, and the higher the concentration of CO2 that you breathe, the more painful it becomes. And people use words like excruciating to describe breathing 100% CO2. There's nothing particularly different about rodents in terms of the neural mechanisms they would have for sensing CO2 as painful. And indeed, if you take an anaesthetized rat and record from a nociceptive pain sensing, in other words, neuron in its nasal epithelium, for instance, they do what the humans describe. They start to respond to CO2 at about 50%, and they show a linear increase in their response in the number of action potentials they fire as the concentration of CO2 increases. So that's a lot of evidence to worry about putting rodents in high concentrations of CO2. That doesn't mean that every time someone kills a rat or a mouse with carbon dioxide, they cause it pain, because there are different ways to use CO2. And most of you will know that one approach to using CO2 is to drop animals into a chamber that's full of 100% CO2, which is pretty much guaranteed to cause the animal pain, albeit it'll die quite quickly. Another approach is to use what's called a gradually rising or gradual fill method, whereby the animal goes into a chamber which is empty, typically, and it's gradually filled with CO2 at something like 20% of its volume per minute, typically somewhere between 10 and 30. And this is recommended and mandated by all sorts of sets of guidelines and legislation around the world. And for a good reason. If you use slow fill, you take advantage of the fact that carbon dioxide is a general anesthetic, and it turns out that you need 30-something% or so of CO2 to get a rat or a mouse unconscious. That's the Mach value essentially for CO2. And so if you use one of these rising fill, gradual fill paradigms, your rat or mouse will likely become unconscious around about high 30% of CO2 or so after a couple of minutes, 2.5 minutes maybe. And so what this means is that the animal's going to be unconscious before it's exposed to those 50% plus concentrations of CO2 that we expect might cause pain. So to go back to my fill curve again, I've marked, so this is concentration of CO2 on the vertical axis versus time on the horizontal axis. And what you can see is that I've marked a grey area, which is where a typical rat would lose consciousness in a 20% per minute filling chamber of CO2. And it takes another minute and a half, 2 minutes to get to that danger zone in red or pink, depending on how your screen looks, which is where potentially the animal could suffer pain if we think the threshold is about 50%. So we can avoid causing these animals pain. So far, so good. But should we worry about using gradual fill CO2? And I'm going to argue that yes, we should. Although we don't cause animals pain, there's significant evidence that there's a danger of these animals suffering stress or distress down here in the left-hand corner of this graph, which is the area before the animal can become unconscious, the first minute and a half, 2 minutes, when it's exposed to low concentrations of CO2. Because pretty much every attempt to measure whether these animals show aversion to CO2 at that kind of concentration finds that they do. And I'm going to show you some of that evidence now. And I think by far the most compelling, certainly the most visually compelling of this evidence comes from Dan Weary's lab at the University of British Columbia, where they've developed this approach avoidance paradigm. So what they've got here in this diagram is 2 rat cages linked by a tube, and a rat who's been taught that he can go down into that lower box to get a food reward that he's really quite keen on retrieving. In this case, it's a pile of Honey Nut Cheerios. And then what you can do is ask what happens to that rat's behavior if I start pumping carbon dioxide into that lower chamber. And what I'm going to do now, hopefully, if we can cue it up, is show you a video which demonstrates exactly what this rat will do. So if we could have video 1 now, please.
So the rat is given a signal and enters the chamber to retrieve his food reward, at which point the chamber begins filling with carbon dioxide. And for a while, this rat tolerates the presence of carbon dioxide in order to get the delicious food reward. But in well under a minute, just over 30 seconds or so, in fact, this rat detects the presence of CO2 and decides to exit the chamber, leaving behind some of the food reward, which would not normally happen if there was only air in the chamber.
Right, so hopefully you saw and heard that video showing that rat showing what I think is quite obvious signs of aversion to carbon dioxide when it was pumped into that chamber. And what he's done is leave behind his food reward, which is something he would never do if you just pumped air into that chamber. So a clear demonstration of behavioral aversion. And in another study, the same group have used fixed concentrations of CO2 to examine the threshold where it starts to become a problem for these animals. And hopefully what you can see is that when the carbon dioxide concentration exceeds about 10%, these animals start to sacrifice their food reward to avoid spending time in the presence of CO2. By the time you hit 20% CO2, you've got no animals exposing themselves to it in return for a food reward. So I think that's reasonably convincing signs of aversion there. It's worth at this point thinking about the alternatives to CO2, and the same group have used that exact same paradigm to look at what rats do when they're exposed to argon or the volatile anesthetic isoflurane in that chamber. And to cut a long story short, what they find is that argon is substantially more aversive to these animals than carbon dioxide, and isoflurane quite significantly less aversive. Some animals spend a lot longer eating their Cheerios in the presence of isoflurane. And it's also worth saying that whenever this group has examined mice, the results are very similar to those of rats. We shouldn't assume that mice are small rats, but in this case they are as averse to carbon dioxide, it would appear, as rats. So does that prove the case? A standard fluffy welfarist like me would say that if an animal avoids something, it demonstrates signs of aversion to that stimulus, and that tends to suggest that it induces a negative affective state in the animal. It makes it feel bad in some way that it wants to avoid. And the extrapolation from that would be that if I didn't give the animal a tunnel to run up, a way of escaping, the likely result would be distress, because the stimulus is both inescapable and the animal lacks a mechanism to adapt to it. Now, you can raise some objections to that. There are some objections sort of in principle, which are, what if carbon dioxide changed your motivational state or changed the deliciousness of Honey Nut Cheerios? Either way, you maybe don't care about being in that lower chamber anymore because you don't have the motivation to get the reward. It's also been suggested once or twice to me that the nematode worm C. elegans, which boasts a total of 302 neurons and probably isn't the most sentient thing you've ever met, can also effectively do a similar task just by travelling down a chemotactic gradient. There are also objections to whether or not there are signs of aversion based on studies which simply look at the behaviour of animals which can't escape CO2 and find little overt behavioural signs of what they suspect would be distress in these animals. And similarly, some studies fail to find physiological markers of distress after the animals have had these experiences. So one study which we did fairly recently in my previous lab, which we haven't published yet, is this conditioned place aversion study, which attempted to examine aversion in a slightly different light by asking the animals whether, not just whether they were averse to these substances, but whether they could remember that aversion and learn to avoid the place where it occurred. And this is really just flipping the place preference paradigm on its head, which is typically used to demonstrate the abuse potential of highly rewarding drugs like heroin and cocaine. Rats and mice can learn to prefer a place where they've been administered those drugs, and it tells you that they're rewarding. Similarly, they can learn to avoid places where bad things happen. So the basic approach to this experiment was to expose a rat to a test substance like carbon dioxide in this chamber on the left, and then on alternate sessions expose him to air in a chamber which is physically identical. It just has some stripes on the wall and a different texture on the floor, so he can tell the two apart. You do this a few times to give him a chance to learn the difference, and then you open up a tunnel between the two and give the animal a free choice about where it wants to spend its time. At this point, there's only air in these two boxes, and the hypothesis would be, if carbon dioxide is bad news, then the animal is going to choose to spend time in the place where it hasn't been exposed to CO2. And to cut a long story short, one slide before that, the advantages of that are twofold, I think. First of all, it relies on memory, so this animal can only make decisions about where it spends its time based on its memory of what happened in those places. When that tunnel's opened up, there's nothing in there to confound its decision based on changes in taste or anything. It can't travel down a chemotactic gradient. It simply has to use its memory for its emotional experience when it was exposed. And the other bonus for this kind of study is that when you train these animals, they're confined in a chamber and exposed to a rising concentration of CO2. It's inescapable. It's exactly what happens to animals when they're killed with this stuff, except that we rescue them after 90 seconds or so, just about when they'd be on the cusp of losing consciousness. And so I'll cut to the chase and show you the results. And basically, the outcome of this study was that in all the substances we tested, we found a strong conditioned place aversion. So we, again, similar to that previous study, looked at argon, carbon dioxide, and isoflurane. And those dark blue bars represent the animal's initial preference for one side or the other of that apparatus, and it's basically 50% give or take. So they don't care which side they spend time on. They're happy to move between the two sides. After they've been trained, conditioned by exposure to these substances, they demonstrate an aversion for the side where they've been exposed to them. They spend less time on those sides. So that's a demonstration that they can remember what happened and avoid those places, and that memory is actually reasonably persistent over a significant period of time as well. In terms of the magnitude of that aversion, in other words, what's worst, well, we agree strongly with those previous results that argon is by far the most aversive of those substances. In terms of carbon dioxide and isoflurane, in our hands they actually came out about equal. They're both aversive, but they're hard to distinguish in terms of the magnitude of aversion. And just to emphasize what I mean when I say profoundly aversive, talking about argon, I'm going to flip back to some more video from Dan Weary, who's kindly shared this one, which is going to show you a rat doing exactly that same task that you saw before in the first video, but this time going down into a chamber that's full of argon. So it's about 98% argon in there. So if we could run video 2 now, please. Okay, so hopefully you can see a pretty dramatic difference with argon, that these animals really aren't prepared to spend any time in the presence of an atmosphere that's effectively entirely argon. It's profoundly aversive. None of the animals ate any of their food reward, and that really corresponds very much to what we found with place aversion. So going back to carbon dioxide, we've demonstrated, I think, quite convincingly that it is aversive. The question is, why is it aversive? Or at least I think that's a question that's worth asking, because it might help us understand what this welfare problem is for these animals. And there are several candidate mechanisms. One area that's often raised, although the evidence is not particularly overwhelming, is that it may be an adaptation to burrowing. These are animals who live in burrows, and they appear to be highly sensitive to CO2, more sensitive than other species. At least rats and mice amongst the rodents appear to be very sensitive to CO2, and that may be because they live in burrows. And we can tell this from a number of studies. There's studies done in IVCs which show that rodents will avoid single-digit concentrations of CO2 when they build up in IVCs. And in terms of mice, we know that they're exquisitely sensitive to CO2. They can detect concentrations way down near atmospheric concentrations. They can actively avoid 0.2% CO2, and they appear to have a neural mechanism for effectively detecting it via olfaction. They have neurons in the nasal epithelium which are connected directly to the olfactory bulb and which are sensitive to carbon dioxide, which is something that certainly humans lack. We would be oblivious to these kind of concentrations, and this might be some adaptation to detecting CO2. So having told you that we probably shouldn't be anthropomorphic about the way rats and mice deal with CO2 because they've got different mechanisms, they may also share some of our responses to excess carbon dioxide. And one of those might be dyspnea, the sensation of air hunger or the uncomfortable urge to breathe. And breathing about 7% CO2 will cause dyspnea in humans. It's a similar sensation to having an asthma attack. And it's very unpleasant, and there's reason to suspect this might happen in rodents. And I apologize, I missed the reference off, but there's a really good review by Nio Bosley and colleagues in the New Zealand Vet Journal, which is really worth reading, raising the question of whether we cause dyspnea in animals we expose to CO2. So that's one area of concern. I think one that there's even better evidence for is that carbon dioxide induces fear in both people and animals. And breathing about 20% CO2 will induce a panic attack in vulnerable humans, people who are vulnerable to panic attacks, in 1 or 2 breaths. Similarly, brief exposure to 20% CO2 in rodents can be shown to activate the panic and defense areas of the brain, bits of brain involved in the fight or flight reflex, the hypothalamic pituitary axis and so on, which tends to correlate what humans experience. And even more interestingly, I think, if you get humans to breathe 7.5% carbon dioxide, so not very much CO2, in a normoxic atmosphere, so you add back in enough oxygen that they're breathing 21% CO2, just like they would be in normal atmospheric air, they feel anxious. And they use all these lists of words that you can see at higher frequency in the right-hand column to describe how they feel when they're breathing CO2. And I think that third one down is completely fascinating. Feel like leaving. They want to get out of the place where they're breathing this stuff. And that sounds remarkably like what those rats going down into that chamber to get their Cheerios decide that they want to do. And I think this really argues quite strongly that what we do when we expose rodents to carbon dioxide, even in low concentrations, is we induce some form of anxiety. And if that isn't enough to convince you, I'm going to resort to some serious molecular biology to try and underpin this point. This is a really interesting study from Zeman and colleagues from a few years ago now, which in mice unpicked both the receptor mechanism and the anatomical substrate for carbon dioxide induced fear in mice. And mice display fear quite often by a freezing behavior. So it's an anti-predator mechanism that they'll freeze when they're afraid. And mice will also display freezing when they're breathing concentrations of CO2 around about 10%. So in this left-hand graph, if you just look at the open, unfilled bars, you'll see that at 10% CO2, these mice suddenly start displaying a lot of freezing behavior. So we've got a sort of potential anxiety response in these mice. And really fascinatingly, what this group did was knock out an ion channel called the ASIC1A, acid-sensing ion channel. And these are the responses of the mice in the black bars in this left-hand graph. And you can see that the freezing response is significantly attenuated in the mice that have had this gene knocked out. So we now know roughly what's responsible for at least a large chunk of this response. It's an ion channel that's responsible for sensing low pH in the brain, which makes sense because carbon dioxide induces low pH, it induces acidosis when you breathe it. Even, I think, more convincingly than that, the same group went on and asked which bit of the brain is the bit that might be responsible for this, and they looked, unsurprisingly, at the amygdala, a bit of the brain that's involved in emotional responses. It's an area that's suspected to be involved in fear responses. And the way they tested this was to inject bicarbonate into the amygdala, and bicarbonate will neutralize the acidosis induced by carbon dioxide, and lo and behold, that attenuates the freezing response in these mice. So we've got the anatomical location and the molecular mechanism for this. So I think putting all of this together, it argues very strongly that even low concentrations of CO2 cause fear or anxiety in these animals and indeed in humans. So I may or may not have convinced you that there's a problem with carbon dioxide. What we really want is a solution if we believe there's a problem. And one commonly suggested solution is pre-anesthesia. In other words, using a volatile anesthetic drug to get these animals unconscious before they're killed. And the Canadian Council on Animal Care has been right at the forefront of advocating this change. They've said that carbon dioxide shouldn't be used where there's a better alternative, and one of their suggested alternatives is anaesthetizing the animal with an inhalant volatile anesthetic. So is that the solution? Well, I've already told you that in those approach avoidance studies, isoflurane and sevoflurane, I think, appear less aversive than carbon dioxide. But I also told you that when I did place aversion studies, it was hard to distinguish the two. They appeared equally aversive. And I think there's some really interesting data from the same lab, from Dan Weary and his colleagues in Vancouver, which sheds some light on why we might have found those differences, amongst other things. And what they did was develop basically the opposite version of their reward-motivated task, where they used a aversion-motivated task. And rats generally don't like to hang out in brightly lit areas where they're in danger of predation and so on. If you put them in a light-dark box like this one you can see, they will tend to, given the choice, hang out in the dark where they feel safe. And so they simply asked the question, what if you pump carbon dioxide into that dark area? Are you going to force this animal to take the risk and go out in the brightly lit area to avoid the CO2, or indeed isoflurane? And the results from that study on first glimpse were entirely consistent with all the previous data from the same lab. So you can ignore the L and H conditions here, they're just different light levels. Just concentrate on the CO2 in green and the isoflurane in the red. What you're looking at is dwelling time in the dark area. You can see that carbon dioxide pushes these animals out into the light area, so they'll take the risk to go and be in what they perceive to be a dangerous place or an unpleasant place to avoid CO2. In the case of isoflurane, they spend a lot longer in the presence of the substance, and some of them even let themselves become effectively anaesthetized. They start to become recumbent. So that all looks jolly good in terms of using isoflurane as a better alternative to CO2. They also, though, did something really interesting. If you look at those second hatched bars that I've just put up, those are the responses of rats who were re-exposed to CO2. They're exposed a second time. And that's not very surprising. There's no change. They're still averse to carbon dioxide. So they don't seem to change their response on second exposure. If you expose rats to isoflurane a second time, there's a really pronounced change in their level of aversion. So they now have become just about as averse to isoflurane as the other rats are to CO2. And I think what that says is that these animals learned something during their first exposure which made them more averse to CO2, to isoflurane, I'm sorry, the second time. So we've probably got some kind of learning mechanism going on. And that explains the difference between place aversion experiments, which involve multiple exposures to train the animals, and tests where the animals might be naive. So I think place aversion overestimates the response because it relies on animals being exposed to these drugs several times to learn the aversion to the place. So in naive animals, it's likely to be lower, and perhaps those approach avoidance experiments are more indicative of the level of aversion, as long as these animals haven't met these drugs before. So they may well be a better alternative, at least for animals that have never seen these drugs before. Of course, quite a lot of lab rats and mice may have been exposed to isoflurane for surgical procedures, and that's a question that really needs to be addressed when we think about killing them, as does something that's well outside the purview of this talk, which is the use of multiple anesthetics for lab animals. So at this point, I'm going to sort of sum up my conclusions about the evidence and then go on to think about what we might do in the future. So really, I would say most of the physical methods are not going to cause an instant death. There is some concern they might cause suffering for short periods of time. There are also procedural concerns and clearly a need for people to be very well trained to use them. Pentabarb, as we've discussed, may cause pain. There are ways that might get us around that, but there are also other issues with handling and the long induction. Carbon dioxide, I think, put bluntly, offers you a choice between a quick, painful death. If you drop a rat into 100% CO2, it'll lose consciousness in 10 or 15 seconds, but those 10 or 15 seconds are not going to be pleasant. Or you can use CO2 in a slowly rising concentration and cause a slow, anxious, fearful death. Aversion to CO2 is, I think, demonstrably significant from those studies and others, and it almost certainly causes fear because we now understand something about the mechanism by which that might come about. And that aversion occurs at low levels where we couldn't get the animal unconscious, so the animal is going to have to experience that every time it's exposed to carbon dioxide. I've shown you that I think for rats and mice, argon is almost certainly worse. Isoflurane may well be an improvement, a significant refinement, especially for naive animals, but there are some questions to think about for animals who have been exposed to this before. So, going back to what we thought about at the start, how are we going to choose the least harmful method? And I think really what I'm going to argue for is that we need to think about these methods really hard every time we decide to use animals. We need to think about how they're killed. Lists of acceptable methods, like we have in the UK and lots of places have, are fine in terms of prohibiting the really, really awful ways of killing these animals. but just ticking the box to say I've chosen one of the list of acceptable methods, I'm not sure is the ideal way. And what we should probably be doing is thinking hard about the current evidence about these techniques and factoring them into the harm-benefit analysis. They're all potential harms, and therefore they should be in the harm-benefit analysis. But we have a lot of unknowns as well, and that's what I'd really like to advocate for, is trying to close some of the gaps in our knowledge about these agents, which would allow us to make better decisions and do better harm-benefit analysis. We know these agents are welfare problems, or at least I think they are, but how bad are they? How bad is being put in carbon dioxide compared to post-surgical pain in a rat, or being a mouse who's too cold because you haven't been given enough bedding? That would allow us to put these in context and know how much we should really be worrying about these issues compared to other issues for these animals. On more specific nature, I think we need to think about understanding more about the response to some of these volatile anesthetics. And even more importantly than any of that, at the end of this, I'd like to emphasize that what I think we should really be doing is looking for a better way to kill these animals. It's not futile to think that there's a better way to kill these animals which might be entirely humane. There are methods for stunning livestock when they're slaughtered for meat, captive bolt stunners, electrical stunning of sheep and so on, that appear to render animals insensible almost instantaneously, allow them to be killed in what many people would regard to be a humane manner. And experimentally, we've seen demonstrations that argon actually, although it looks horrible for rodents, might be quite a humane way of getting pigs unconscious. And we'll come to that in a second when we queue up the very last video. And also we've seen recent evidence that fish are less averse to some novel agents than some of the traditional anesthetics that are used to kill them. And I'm now going to show you why we need to consider species when we look at different ways of killing these animals, by showing you very quickly the response of a pig placed in an experiment where he's exposed to argon.
So this pig is doing his version of the Cheerios experiment, but this time for a reward of apples, in a chamber that's effectively almost free of air, contains about 98% argon. And as you can see, he shows no detectable sign of aversion, and in fact lets himself become rendered almost unconscious. And just to prove that there's no signs of aversion, the pig goes back for a second attempt.
Okay, so hopefully what you saw there is that pigs appear, at least in an experimental setup, to be perfectly happy to expose themselves to an atmosphere that contains almost no air. And I'd like to thank Mohan Raj, who shared that video and did some really pioneering work looking at this back in Bristol. And I think that just really says there are methods that we should think about that can be humane for certain species, and why can't we find them for rodents? Here's a few random thoughts about what they might be. Devices to mechanically assist cervical dislocation to make sure it's more reliable. How about some physical stunners for rodents? In the wild, we have this good nature trap with the little diagram there, where a rat goes and sticks his head up a baited tube and trips a little trigger and gets shot in the head by a captive bolt. The last thing that rat knows about is being incredibly pleased that he's found some delicious food. That seems probably like a better way to go than some of the things we've talked about. Low atmospheric pressure stunning has been suggested for birds and pigs. It might work for rodents. Someone should certainly check. It may be that rodents care more about hypoxia than other animals, but it's worth looking. Maybe we can find novel chemical agents, either injectable agents, rodenticides, who knows. Rats have been killed in labs, and other livestock species have been experimentally killed using focused microwave irradiation. It's worth looking to see if that's humane. And who knows, there's got to be something out there somewhere. Just the very last slide, because it always comes up. People say, I'm still using CO2 or I'm using isoflurane. What can we do in the meantime to refine it? Well, one thing that comes up quite often is a little bit of evidence, but not much, to suggest it's an improvement, is to kill animals in their home cage, take all the procedural stress out of the equation by not taking them out, handling them, placing them in an unfamiliar environment. There are all sorts of other refinements, like a study we did showing that adding nitrous oxide can get animals unconscious faster when you're using carbon dioxide. There is clearly an optimal fill rate as well, which hasn't been found yet. It might be that 20% isn't the ideal fill rate, and you can get animals unconscious faster without causing pain. I think all of those are only refinements, though, and there's still going to be issues, but it's worth being aware and trying to refine your current practice until we can replace these agents. And I will just at the end conclude very quickly by saying I think it's hard to understand whether animals' deaths are humane or not, but we've made huge progress. There's some amazing studies there and lots of others that I haven't had time to talk about. We need to put this in the harm-benefit analysis because all of them are a welfare problem, and we need to find a better way of doing it. At the very end, I'll thank the NC3Rs who supported the research in my lab, alongside you four who supported me even before they gave me a job, and Charles River, who also supported some of the research, and some of my colleagues. And at the end, I will leave up this slide just with some links to other sources of information if that's of any use to anyone, and take some questions. So I'm going to let Steve pick out the hard ones for me.
Well, thank you, Huw, for that really informative presentation. Before we get started on the question and answer session, I'd like to remind you, the audience, on how you can submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left on the presentation window. And we'll try and answer as many questions as we can in the 10 minutes or so that we've got available. So, our first question, if we're going through, Deb Hickman at Indiana University asks, the induction of anesthesia is distressing. So would the animals not have suffered during induction of anesthesia prior to a chemical overdose or physical methods?
Well, that's a good question. I don't think we understand what these animals are using to learn that exposure to isoflurane is distressing. So something bad happened that first time that they're exposed. But bear in mind that these animals also had to recover. They were exposed, they went into that chamber, they were exposed to isoflurane. They then had to go through the recovery process. So it's unclear whether it's the induction or the recovery or the ongoing exposure when they're walking around in that chamber, which is the problem. So I think we're not certain yet is the answer. They don't display immediate signs of trying to avoid it on the first 30 seconds or so of induction, so it might be more humane, but I agree that there are some concerns, and certainly we need more experiments, and I suspect isoflurane isn't perfect at the very least.
Deb Hickman's also asked another question too from Indiana University. Please comment on EEG and consciousness as compared to reflexive response to pain for decapitation.
Yeah, I mean, a good point. Neither of them are particularly brilliant measures of consciousness, because we can clearly record that EEG in an unconscious animal which is anaesthetized, so we can't be certain that it would have been conscious. And you can have reflex responses when you're anaesthetized too. So unfortunately we don't have a neural signature of consciousness which would allow us to unequivocally diagnose whether a decapitated rat is still conscious and perceiving what's going on, let alone whether it's in pain. And that is a real problem, I think. So I simply raise those ongoing EEGs as something to worry about. The absence of an EEG would be much more reassuring that the animal was unconscious or dead.
Question from Mary Wick at the Fred Hutchinson Cancer Research Centre. Please clarify euthanasia by concussion. Is another method employed following the concussion?
Yeah, absolutely. Sorry, I should have said that. I think typically people would do cervical dislocation, decapitation, something like that. Yeah, absolutely. I should have said that if I didn't.
Lynn Nivrana at AstraZeneca says, can pentobarb be administered via the rat tail vein instead of IP for a quicker death?
I would have thought so. I've certainly done that in anaesthetised animals who already had a tail vein line in place. I think the worry is just that lots of people don't have the skill to get a tail vein line in an animal, particularly one who's not particularly compliant with being handled.
Deb Hickman again from Indiana University. Very low flow at approximately 20% is what you're focusing on, aside from fully charged chambers. What is your opinion on gradual fill chambers, 40 to 60%? There's some literature, such as that by Dan Weary, that says 50% may be better for mice.
Yeah, I agree. I think I touched on that at the end, that we certainly can look for the optimal flow rate that'll get these animals unconscious as quickly as possible without touching on the areas where they might be in pain. And 20% was a somewhat arbitrary guess on what might be a good fill rate. I just still think it will take a significant amount of time to get those animals unconscious, and they're not going to avoid feeling anxiety during that. But I agree, and I encourage attempts to find the optimal fill rate.
Question from Agnieszka from the Institute of Industrial Organic Chemistry. Can CO2 be used in toxicological studies? Won't it affect the gross and histopathological examinations?
It may well do. It certainly causes signs of bleeding in the lungs sometimes and so on. I'm not a toxicologist or a histopathologist, so I will defer to their expertise, but it certainly could be a confound in some studies, and you may well need an alternative.
Jodie Gastrich from Mercy Health says, have there been evaluations of additives to CO2 to counteract the negative sensations or perceptions?
So there's a study by Valentine and colleagues looking at the sort of standard anaesthetic pre-meds, and really in their hands they didn't find any great improvement. And of course, generally what you're talking about then is injecting the animal with a drug and handling it and stressing it out by doing that before you kill it with this other stress-inducing agent. So that may not be the way around it. The other thing that's often thought about, of course, is what if I add oxygen to keep the animal normoxic and just kill it with the CO2? And I think Dan Weary and colleagues found that that really didn't help, that it does seem to be the CO2 per se rather than the hypoxia that's the issue.
Argo Zakadraka from the Experimental Research Centre at Elpan Pharmaceutical Company says, do you have any data regarding the use of sevoflurane compared to isoflurane?
So again, I'm going to point to Dan and his colleagues at UBC who did look at Sevo. I think generally the responses are quite similar, and that does include development of stronger aversion when you've met it more than once.
Tim Connold from the Animal and Plant Health Agency in Weybridge, in the UK, says, regarding the aversion test, I can understand the aversion to CO2, but could the aversion to isoflurane not be caused by the smell, which may not necessarily be stressful? Would it be better to have a controlled experiment where the controls are rodents that are exposed to air with a strange but not harmful smell?
Well, I think some of these studies do include, certainly some of the approach avoidance studies have used things like menthol. And I don't think it's the smell per se, but I strongly suspect that what they're doing is learning that the smell is the indicator that something is unpleasant is going to happen. So in other words, we've got a kind of Pavlovian response to the smell, and that would what would make isoflurane easy to learn about, that the smell will come on long before the effects that we suspect might be what's making these animals averse to it, which might, I suspect, be more likely to be nausea given the human response to some of these drugs.
Lillian Gomez says, according to the Bailey study mentioned previously, 7.5% of carbon dioxide is anxiogenic in humans. Is that an average of a study with a specific group, considering divers and the treatment of panic and conversive syndrome at the emergency room using a bag given to the patient to breathe in instead of BZ-DEDs and SSBIs?
So I think, as I remember, it's a while ago, that study is looking at people who are naive. It is an average, and you're just looking at their percentage changes, the up regulation of all of those reports, self-reports of how they feel whilst breathing 7.5% CO2 from a bag. And it's over a relatively prolonged, as in minutes, exposure. So it's not the sort of single breath stuff that causes a panic attack at 20%.
So Damien O'Donoghue from Prospect Solutions says, I don't mind being drunk as much as I hate the subsequent hangover. So in the conditioned place preference studies, how much of the animal's displeasure is derived from the exposure to the test gas, and how much is due to a post-exposure effect?
Yeah, well, I think we just basically covered that, and I also took out the slide of the mouse holding a tiny little bottle of vodka, because that was one of my hypotheses too, that it may well be the recovery from some of that anesthesia that's the problem, and that really needs unpicking. And I agree that that's a really good hypothesis.
Anna Valentin from IBMC says, do you know if there is any discussion to review the European directive regarding euthanasia, taking out argon and nitrogen as agents to be used in rodents?
I'm not party to the discussions in Europe. The UK didn't transcribe that part. They left out the inert gases. Unfortunately, we also took them out for the farm species, I think. And it clearly strongly argues for a sort of species-specific consideration of that inclusion, because it's justifiably included for some other species.
Looking at the time, I think we've only got time for one last question, I'm afraid. There's lots have come in, so we've just had to sort of pick through them. Final question is from Marcel Perret-Gentle. Said, excellent discussion. Thank you for the powerful insights. So in light of the presented evidence on the use of CO2 for euthanasia of rodents, what are your thoughts on the AVMA guidelines of a 10 to 30% air displacement rate?
Someone was bound to ask me that. I'm not sure we've got the evidence to say that that's not the best way to use CO2 at the moment, but I would keep in mind, as Deb raised earlier, the flow rate, a higher flow rate might be better. And it's almost a philosophical judgment as to whether you prefer pre-fill and a quick painful death to a slow anxious one, which I haven't solved in my own mind, and to be honest I would prefer to not have to think about it by finding a better way to use CO2. But I think there may be some more studies around which will clarify what might be the best of a bad bunch of flow rates, as it were.
Okay, well, I'd like to thank Dr. Huw Golledge once again for his excellent presentation. Before we finish, any final comments you want to make?
Thanks a lot for listening. There's my email address there if you want to send more questions. I think you can send questions by lab routes as well, and I'm more than happy to answer more questions or provide more info. But thank you very much for virtually listening.
Thank you. And just to finish, today's webcast will be available for on-demand viewing for the next 6 months until July 2016, and you'll receive an email from LabRoots alerting you when this webcast will be available for replay. So we invite you to forward this announcement to your colleagues who may have missed today's live event, and thank you very much for watching. Thank you.
Thanks.